very much, Peter. Now, Michelle and the other gentleman at the end there, another Peter, of course, have joined us. We're not going to ask you, I'm going to ask you questions, though. So, Michelle, you know, we've made tremendous strides on the productivity in the Holstein breed. Maybe you could comment on the new emphasis on the workability traits, the health traits, and why that's important in terms of a social license to operate in many respects.
Yeah, so you know, I already tried to squeeze too much in my talk, but you know, it's like if I was telling the whole story through this productivity, but in fact, you know, one of the nice things about the methodologies that existed and its inherent property of what is being developed, genomic selection, is that they equally apply to whichever trait you're interested in, as long as there is a certain degree of heritability. And it's clear that at the present time there is a shift in emphasis going away from pure productivity to a certain number of other traits, which are, you know, pushed upon, I would say, by a number of certainly justified societal concerns. In the end, I think it's the economics that will dominate. But so the nice feature of these methodologies is that, you know, they just apply to whichever trait or index that is a combination of traits that is considered at the present time, at a certain time and place to be the selected breeding objective.
Good. Peter Langridge, can you give us, you touched on it briefly, and then I'm going to get Fuzi and perhaps Isobel to talk about, we've just recently decommissioned the wheat board here, and I sit with individuals and say that's a great opportunity, and then I hear others that say it's going to be a disaster. So in terms of plant breeding and the private initiative and competitiveness of private individuals, where do you see, what's your opinion with respect to this move in Canada?
For an Australian, there's a degree of deja vu in that we've already gone through part of this process in Australia. Our marketing authority, the Australian Wheat Board, that was essentially disbanded because they were involved in providing bribes to various officials in Iraq. So,
well, you are a bunch of fugitives.
Yeah.
A bunch of fugitives down there.
For the researchers, it's become a bit of an issue because for a crop like wheat, the quality of the grain is critical. You know, it's used for making bread, various other things. It doesn't matter for corn, you know, just for the starch basically. So the quality is not an issue. So that's been a big constraint on breeders. We've relied very much on these marketing authorities to give us information and intelligence about what our customers want. So for both Canada and Australia, we export most of the grain that we produce. So we need to know what our export markets want, what their key quality requirements are, and feed that back through the breeding programs into the research activities. And with the loss of some of these marketing authorities, it's now falling to the breeding programs themselves to try and generate much of the market information. And that's actually an expensive and difficult task. And I think and I suspect it'll happen in Canada as well if it hasn't already happened. There's a danger of a disconnect between the research breeding activities and our market requirements and expectations. And that needs to be addressed.
Okay, thank you. Fuzi, would you care to comment?
Sure. I mean, in our program, we're focusing on improving yield while maintaining quality, and Canadian wheat is known for its high quality. Now, in the future, how that will affect the future varieties, it's difficult to say, but as other players comes in, and probably there will be a shift maybe in the proportion of certain classes of wheat, and again, based on the demand from the industry and the players that are going to play a role now instead of the Canadian Wheat Board.
Isobel, make a comment on that and what direction you're going in with canola.
Well, in the context, canola has been in the private industry now for a long time. There's very few public breeders left in canola. And so we've already gone through this process. But there is still a need for investment in public research, basically, for canola. And a lot of that is funded by the producer groups. So the producer groups have a checkoff where they put in money for research, and that gives them the ability to direct some of the things that they're looking for in the field, because they don't necessarily think that the seed companies represent their interests, because basically they're the seed companies' customers, and so they don't think they're getting the best bang for the buck from the company, so they turn to some of the public institutes to provide research that directly for the producers themselves.
Okay, thank you. I think we'll open it up for questions from the floor or tweets. Who's monitoring the tweets there? Are there any? No? Oh, down here. Okay. Does anybody have any questions yet? David, the head of David Bailey, Genome, Alberta.
Yes, thank you. First, a comment and then a question. Comment to Michelle, thank you for a real clarity and a very tough presentation on the topic, so that was very helpful. And my question is, in all the Genome Canada funded projects, there's a GELS component. So for the livestock people on the panel here, what areas of GELS or societal issues do you think in the livestock we're not addressing right now, and we should as we go forward? You've talked about the demand for food, increasing animal protein in the diet for the large numbers of population to increase, but there's lots of societal issues I think there, and I'd like maybe one or two suggestions from Peter or Graham or Michelle on that topic.
Well, I'll start with Graham first, and then Philip would like to weigh in as well, and anyone else, put your hands up, please.
Okay, gosh, I think the two things that immediately come to mind is, one, as I think Michelle said very eloquently, is, this technology can be used for all of the traits we may, that are genetically, have a genetic component. We can use them for all of those. And I think working with people like Peter and my colleagues in Alberta is using those approaches to find out what traits are going to be important and what people's willingness to pay for those traits are and what the balance is and how we incorporate those into our selection indexes. I think that's really important. And I think the other thing which I'm not convinced funders, other than Genome Canada, necessarily get is the importance in that research in terms of looking at barriers to adoption. And we have examples of technologies, plenty of examples of technologies that get thrown over the wall and don't get taken up. And I think we need to be more proactive in looking at what the technologies are, what the barriers are going to be, and do it in parallel. I think that's really important.
Philip?
And let me amplify that. The discussion about GELS generally seems to be framing it as a question of ethics and what we should and should not do. In fact, if you look at the activity in the GELS community in Canada right now and internationally, the bulk of the work is no longer talking about the ethical dimensions of the application of technology, but it's looking at the models, at the hurdles that are out there in terms of creating and using technology in a socially acceptable way, and then developing different institutional choices that may improve the uptake and use. The real issue in a lot of cases isn't the cost of compliance with marketing or with the regulatory system or others. It's the time that it takes to go through the system. Time is the killer for most of the science that we're talking about here. Partly it becomes a little bit stagnant and it isn't as competitive anymore, but in terms of return, it diminishes the return very rapidly. A 1 or 2 year lag it can kill it. So that the real issue around GELS these days isn't about whether it's ethically and socially acceptable or not. That's certainly a consideration. But do we have the institutional mechanisms, the tools, the structures that actually can take a good innovation that adds value either in the marketplace or in other uses and brings that into uptake and use in society wherever it's most appropriate?
I'm going to ask Willie to comment because you're right in the heartbed of Lower Fraser Valley. My goodness, that's where consumers rule. So, from David Suzuki country, how's your view of that question?
My blood pressure has just gone up. Canada is a highly polarized country when it comes to aquaculture. If you go to the East Coast, you would probably be hard-pressed to find anyone who didn't value aquaculture as an activity. If you go to the lower mainland of BC, on the other hand, and you go to a party and you mention you work in aquaculture, you might as well be wearing a fur coat. You may not get out of there alive. The country is so polarized. When we go to international meetings and we see aquaculture representatives from Canada, there's usually 1 or 2 from BC, and there's probably 50 or 100 from Eastern Canada. It is that polarized. And so gels, it's an incredibly difficult task to understand what is it that makes people in different parts of the country have so different views on the value of food production. And I have no idea.
Well, I'm glad I asked you for a reason. Okay, this gentleman over here. And
So
maybe
the-
I will ask, because I certainly don't know your name, I apologize. Give your name, who you represent, and then the question, please.
Ken Knox is my name, and I'm on the Genome Alberta board,
I should have known
resident
that.
from Ontario. So the fur coat story brings my question. So last night, coming back from the wonderful dinner, I was cold, and I got thinking the human being could have genetically been modified so that we can sustain the cold, but instead we wear coats. I didn't have mine. My question comes then to all the discussion we've had on the plant side today. We talk about genetically modifying the seed. We haven't talked about coatings, and yet we know there's an awful lot of work going on in the area of coatings, and I'd like a comment.
Okay, who wants to volunteer? Okay, Peter?
I'll go for it. You know, plants, of course, have a real problem in that they're stuck out in the environment, and they've got to take whatever the world throws at them. And I think that's one of the reasons why plant genomes have become so complex. They've developed quite sophisticated mechanisms for dealing with stresses and coping with stresses and managing the interactions between stresses. And the opportunity to select and improve those is definitely there. I think we need to be aware that most of our major crops are now being grown in environments where they didn't evolve, and therefore we need to sort of go back and re-engineer or redesign the plants in many ways. And I think that's what many of the groups around the world are now doing. And the results are pretty encouraging, I think, for most of the work that's going on.
Okay. John, what do we have, 5 minutes? Okay. Just a question I have, and I'd like all of you to comment quickly. Where in your own area of research and activity, identify the biggest gap, and I think I know what you might say, Graham, but you get a chance to be on a pedestal here. It's so vitally important that research continues and the drive towards commercialization and the partnerships that go on, but foundationally, what are your biggest challenges? I don't simply want you to say funding. I mean, that can be a common thread, but it might be. And then I want to, because I'm putting this I'm going to frame this with my feeling that communication is so critically important. And I know Christina was running around here getting the messages out on YouTube and the web. And so my feeling is that we need to get every Canadian, everybody in the world, to put their arms around genomics and this type of reach, because it touches all of us. And, you know, the One Health concept, or the intersect of animals, humans, the environment, all affected. That's been well stated. So I'd like each of you to say, you know, if you had a wish list and the number one thing, the inference might be funding, but more specifically, what that funding would give you. So Graham, I'll start with you, and we'll just go down the row. And somebody needs to be taking notes and filling those gaps there. So
I guess if I only have one, it would be, and I think we heard it throughout the event, is it's about phenotypes. I think even this group has also said it. We need to drive the technology. You know, we really are in a fabulous position now in terms of technology. Okay, we can combine some of the technologies and do even more. But genomics isn't the issue. It's actually getting the phenotypic information, large enough populations or samples and using that to drive the technology. That's the biggest gap.
Okay.
I would agree entirely with that. Getting the phenotype is actually very, very complicated. And having facilities where you can get a true phenotype is actually a big problem for not just salmonid biology, but I think all fish production. So how do you actually do a challenge test, for example? A challenge test with a virus. How do you characterize the virus? How much of the virus do you give? If you do work with sea lice, what stage of the sea lice do you put in? How do you know there's no other effectors going on? How do you control salinity? How do you control temperature? All of these factors will play a role. And I don't think in Canada, I know in Canada, we do not have a facility where you could, which you could rent for a month, 2 months, 4 months, to actually carry out those experiments. I just came back from Chile about a month ago, and they've just spent close to $10 million building such a facility. And we will see the return on investment, which is a big term, but a return on investment in the Chilean aquaculture industry. And it's not just going to be for salmon, it's going to be for tilapia, it's going to be for you name it. They're going to do it.
Very good. Fuzi?
So I will mention a couple. I mean, phenotyping is definitely an important parameter and factors we need to look at. But within the wheat, I will mention bioinformatics and breeding. I mean, bioinformatics is because, I mean, the technology in sequencing has been developed so much that now we can generate sequence easily. But it's the analysis and the interpretation of data. And we need to pay more attention to that and ensure that we have the resources able to do that. And the next one is breeding. Conventional breeders have done wonderful jobs over the years in improving varieties, but the future breeders need to look at and capture some of the information now that coming through genomics and translate that into the field. And we have to keep forming and training breeders to continue doing, to adopt this new technology. And I believe that will be the future of breeding.
Isobel?
Well, you know, I'd echo a lot of the previous speakers, and so phenotyping is a big issue with regard, particularly some of the stresses like the cold and the drought, which are very complex and very hard to replicate under lab conditions. But the other thing that we're facing is that we're developing other, sort of trying to diversify the crop in the prairies, so we're looking at other species. So for example, Camelina sativa has a potential role in bioproduct platforms, and also actually in the aquaculture industry, we're looking at using it as a fish meal product. But the problem we're going to face is that the regulatory process is going to be so slow in being able to get these crops into the Canadian agriculture and sort of more widely accepted. And so I think that's going to become a problem for us.
Well, you've got a fellow from CFIA, maybe not in that division, but right beside you.
I actually, interestingly, I can echo a lot of the things that you guys were talking about, even though my outlook is so different. We have, certainly, as you were suggesting, bioinformatics is definitely a bottleneck right now. Generating the sequence data, however reliable or unreliable it may be at this time, is not really a big issue. We can even do it cheaply. You can do a bacterial genome now, I'm told, down to about $1,000, and reasonably we can expect it to be down in the hundreds in no time. And I think in a public health emergency scenario, that's quite justifiable expenditure. The annotating, finishing a genome obviously takes days, if not weeks, to do, even for a bacterial genome. However, it may not necessarily, it may not be necessary to have that degree of completion of a genome to pick out some salient markers to do a traceback investigation, but that remains a bottleneck. The other thing is we can, I think, reasonably expect that with genomics in our area, we're going to identify a multitude of different gene markers that we would like to interrogate. If we have a bacterial isolate from a food or a clinical sample, we'd like to interrogate to determine whether or not it has this or that marker. We need better platform technologies that can accommodate multiple gene markers. So multiplex, high throughput technologies are going to be required, and I don't think we're there yet. We have some candidate technologies that could be going in that direction, but we're some years away. And then finally, this issue of phenotyping. In our case, I was mentioning how we would use the bacterial blueprint as the ultimate index or basis for profiling. For example, we want a virulence, we want to profile, is this isolate more virulent than another isolate? And we would do that on the basis of looking at a number of, say, virulence genes. So supposing you can detect a toxin gene, does that, though, in fact, necessarily correlate with the expression of that toxin? Is the mere detection of a DNA sequence necessarily a reliable indicator that this particular isolate can produce that toxin? And I think there may be ways of looking at genomic information to determine whether or not a gene, one might be able to reasonably predict if a gene would be expressed. But again, we're not quite there yet. So these are areas that I think research would have to address.
Good. Peter?
I think one of the big challenges that the Canadian part of the business is facing, and it's not a uniquely Canadian problem, has been the shortening of the horizon of planning and investing. That's not a new phenomenon in the industrial side, but in the public side, we're changing the rules of where the public sector works and how long their horizon is. And in absence of somebody having a long-term energy in a sustained program with real goals, a lot of things happen, but they don't happen in ways that connect to the market and connect to the uses. So I think there's a lot more we need to do about tailoring the public-private divide, and it won't be one size fits all. It'll be different in fish and different in canola and different in corn and different in wheat. But getting that balance right, I think, will be critical to realizing many of the technological innovations that people are talking about here.
Michelle?
So, I was just thinking about probably quite differently, but I think one of the things that I would maybe experience as being one of the most important limiting factor in the progress that we can achieve in animal genetics, and I think that it might apply to agricultural science in general, is the image that this field at the present time has in society and the attractiveness that it has for young and brilliant people. So I think we have, for I don't know exactly what reason, the image of our science in society has declined, I think, quite dramatically over the last 50 years. And although, don't take me wrong, there are very young, very smart people that work in our fields, but many of the brightest today would not choose to come and work on agricultural topics, while in fact, like you were saying before, it looks like a very important problem to devote your time or your life to. And I think at the present time that doesn't work very well, and it's one of the limiting factors. I think we have to work on reshaping our image to make sure that the brightest brains in our societies, and that's especially true for Europe, I think, happily come and work on these topics.
I identified communication and branding, and that's all part of it. So there we go. Peter, you came the furthest. You get the last word, and we'll wrap it up.
That's right, and most of the comments have already been said, but I really was also thinking along very similar lines. How do we get community to change their attitudes towards food, to make good decisions, appropriate decisions about food, not based upon mythology, but based on real quality? And related to that is, how do we engage the other components of the food sector? We all here work essentially on food or on the commodity that goes into food. The value generation, though, in food supply is in the processing, the manufacturing, and the retail. They contribute nothing to the development of high-quality food. And this is in stark contrast to what's happening in the medical area, where there's a pharmaceutical industry that invests massively in research. We are stuck there producing a commodity, having to build our research capability, build our innovation around the commodity, not around the full path. And I think the trigger for that would be a change in community attitude, recognition of the value of food, the quality of food, what goes into producing the food, and being willing to pay for it properly.
Great. Well, with that, I want to thank all the panelists. Very insightful, very engaged, many bright minds in this room and also up here. So let's show our appreciation, and thank you very much.